full oxygen methylation or alkylation, except that it would be a carbon center that would be attached, so that a CH3 group, for example, could be attached to the ring. It turns out that often it's attached in an activated position between a couple of phenolic groups. But in any case, methionine methyl would be the usual methylating agent, and the isoprenyl pyrophosphate would be the usual attacking agent in the case of the addition of an isoprene type side chain. So electrophilic units would attack, and the nucleophilic center would be the site attack, so that this center here would be nucleophilic, the carbon of the ring would be nucleophilic, and the attacking group would be electrophilic, and that would enable encourage the reaction. We'll talk more about what that means later, so don't worry if these terms seem a little bit less familiar to you. It's been a while since most of you have had organic chemistry, I realize. Coming then to the degradative comments, this has only recently begun to be studied in any appreciable degree, but I think it's quite important. It's always been assumed that it's not important. The idea has been that lignin, for example, is a terminal product, and once formed, lignin is quite insoluble and it's not likely to be metabolized. But if you think about it, there are instances where lignin must be metabolized. For example, the more obvious and large-scale example would be, say, in a coconut. If you've ever had occasion to try to crack a coconut, you realize it has a pretty heavily lignified hard shell, and yet the seed must sprout. Well, if you've ever watched or dug up a few sprouting coconuts, you'll find that they don't split open as you might expect. You might think that they take up moisture and crack open like a nut. No, they don't. They dissolve the hole and sprout through the hole. So clearly there has to be an enzyme system, presumably, or some mechanism to metabolize the lignin at that site. Now, it is also true, while we're giving you some Polynesian background, if you want to get into a coconut and don't have any easy way to do it, one of the little monkey face that has two eyes in the nose or two eyes in the mouth, whatever, one of the three, if you take an ice pick, is a good deal more easily penetrated than the other two, so that and that's the one it'll sprout through. So it's not entirely obvious that it's a matter of dissolving lignin, but I have taken some time to beachcomb a little and break into a few, and the hole gets definitely bigger without cracking, so that there clearly, it seems to me, has to be some digestive effect. And in other seeds, grape seeds for example, they also sprout from inside a heavily lignified tissue, not always by cracking it open and just splitting their way out. That's a fairly trivial example, I think, but if we take some more important examples, it has been shown that tannin, for example, there are tissues, and we'll talk about this later, but if this is a group of plant cells in some plant tissue, some of these cells characteristically will be heavily full of tannin. These would be sometimes called idioblasts, and we'll talk what that means and why, but in any case, some cells in plant tissues that are fairly high in tannin usually contain a great deal of tannin, so much so that you can see it under the microscope just in ordinary light even as a heavy brown deposit, whereas the adjacent cells have little or no tannin. Well, it can be shown, and has been shown by tissue culture studies, that these cells can in fact revert from the apparently dead, completely saturated with tannin condition to a meristematic or cell-dividing condition. And in the process, the tannin disappears and the cell then is able to go through mitosis and so on. This being so, clearly there has to be a mechanism that can reverse the deposition of tannin and, as we suggested, probably lignin, and these would be the least likely to be degraded, I think, among the phenolic substances. If we look at more simple and more soluble phenols, then we do find that the rate of metabolism it can be high in some instances, and certainly degradation must occur. I was fascinated by a little when we were living in South Africa on our only sabbatic leave, we had a little bush in the front yard, and I asked the neighbors what the name of it was, and they said the local name at least was Yesterday, Today, and Tomorrow, which was intriguing, and I couldn't wait for it to bloom because they explained that the name was based on the fact that the little flowers, when they were ready to bloom but not opened up yet, were quite red, and so that would be tomorrow, and the ones that were opened up would, for this day would be blue, so you'd go from red to blue, and then yesterday, the day after they bloomed, the same flower would be there, but it would be white. And it was rather an intriguing plant because it was a little shrub, and all three would be present at all times, so that you could see red, blue, and white flowers, and it wasn't that the flowers differed, but it was their time of life that differed. So clearly, if you're going to convert a red to a blue pigment and then convert it back to a white, there has to be some metabolism going on. And clearly from the blue to the white stage, it would be likely to be a degradative type reaction. It wouldn't have to be. I could argue that it is degradative from some of my own work. I did a little work with pineapple leaves, which I might as well bring in now because we need a little application date at this point, I think, as compared to too much factual. I just tabulated. On a pineapple leaf, if we look at the leaf, the surface is quite red if the sunlight has been intense. If you grow the pineapple in a greenhouse or if it's been cloudy weather, then the leaf will not be red. And so I hit on the idea, let's try to use this as a measure of sunlight. So we may use the leaf itself as a sunlight sensor. So I tried to see if it would work, and what we did was cover the leaf with some papers and aluminum foil and so on. If we do a leaf in cross-section, the pineapple is a good one to study because the red is confined to an epidermal layer of tough cells, and then just below this is a layer of long chlorophyll-free cells that is called the water storage tissue. And if it is that, if the plant is under terrible water stress, those cells shrink down and are hardly seen. But if there's plenty of water, this water storage tissue is big and juicy, and then the chlorophyll-bearing tissue, the firm leaf tissue, is below that. So it makes it easy to study the anthocyanin-bearing tissue. It's all confined to this tough layer here. All you have to do is stick a pineapple leaf in the deep freeze, and then bend it over a sharp edge, and that'll just peel off as nicely as you please, and you have the red layer completely separated from chlorophyll. Or you can take, as we usually do, a cork borer plug and peel that off and drop it into a test tube of alcohol and acid and extract the anthocyanin. So you have a measure of how much anthocyanin is present. Well, to make a long story short, when we put a band of light-absorbing material over the leaf, we found that yes, the anthocyanin, and we knew this to begin with because what started us interested in, as a matter of fact, if in the field you find two leaves crossing and the one that's underneath in the shade will lack the anthocyanin and the tips out on the outside will. So when we covered up the leaf, it took about 7 days for the anthocyanin to disappear. Clearly there was a metabolic reaction going on, presumably a degradative reaction. And the way we showed it was in fact degradative was that if we used aluminum foil so the leaf was completely dark, it turned out that we had, when we dropped the leaf section would be absolutely colorless and no visible anthocyanin, but in acid solution it became red again. So the anthocyanin was being converted to some form of a readily converted leucoanthocyanin, so you wouldn't really call that a degradative reaction. But if, on the other hand, we used red cellophane, so that the red cellophane apparently took on the duties, whatever they were, of the red pigment in the leaf, then we got fading, it was colorless, and we got, in fact, complete remaining colorless, regardless of treatment. So we didn't have a leucoanthocyanin production. In fact, we had a degradation, or at least a complete metabolism, so that no longer did we have some regenerable form of the anthocyanin. We may come back to that in terms of light reactions and some of the functions of these pigments, but for the moment I think that covers an idea that there is a degradative reaction going on. Among the relatively few studies made in this area is the paper cited in your notes by Proctor and Creasy, where they worked with the loss of color in chicory flowers, and chicory flowers do something like the yesterday, today, and tomorrow, in that they are blue and then they fade out to white. And this has been shown to be catechol requiring catecholase oxidation, so it's a degradation, and they didn't follow, it's an oxidative change, and then the products are further metabolized. They didn't follow what is the ultimate result of this, but probably, if we skip to the bottom of the page, and I hope your notes aren't too badly cut off, we know from microbial studies that the usual metabolism among fungi of phenols is to, with a monophenol, is to add an OH group. So the OH group would be added, starting with 1, and it would be hydroxylated to give 2, and then the compound is oxidized between the two OH groups to end up with cis-cis muconic acid, which would be the acid, dicarboxylic acid with the 2. Now the double bond would have to shift, with the 2 double bonds arranged in di-cis form, so that you can see the rings still exist, and then of course that could isomerize and would be metabolized like any other dicarboxylic acid as a fatty acid from one end or the other, at least down to the form of either formic or oxalic acid, depending on what would be the terminal result. And then these could be metabolized by other mechanisms. As far as we know, if aromatic compounds are going to be broken down in plant tissue to non-aromatic aliphatic compounds, they go by this route. And this would mean that the catechol oxidase participation would make good sense. It's been shown that if you administer tyrosine to, what was it, pear, was it, I think? It's in your notes, and I don't see it right this second. Pear leaf, right, all right. Administration of radioactive tyrosine to pear leaf, then a reasonably high proportion, something over 33% of the total amount distributed, became aliphatic. So clearly the aromatic ring was being degraded and did appear in new products. Fairly recent work, and I don't have the reference right in front of me, but in the last year there's a paper that shows, I think we mentioned already, in the case of the flavonoids, if you introduce fluoroglucinol into the system, it is not incorporated into the A ring. So even though the A ring is a fluoroglucinol derivative, it's built from acetate. And more recent work shows that additional studies along this line, that if they added radioactive fluoroglucinol, they did find radioactivity in flavonoids, but by further study they were able to show that this was by complete ordering of the carbons. In other words, the fluoroglucinol gave sugar, it gave anything else you want to name. The fluoroglucinol was metabolized as an acetate, and apparently an easily available acetate form, so that the plant was able to reverse the construction of fluoroglucinol from acetate to generate acetate, and then that was used to make whatever else might have been being prepared at that time, including carbohydrates and other compounds, and flavonoids for that matter, but certainly was not incorporated directly. And a final example, which is to me the most startling, and I think although the tea plant is quite different from a grapevine, tea belongs to the camellia family and is very different from a grape. Nevertheless, there have some similarities, and there's some similarities in the beverage because like several beverages of this kind, they have a high tannin or high phenolic content. So a lot of good work on the phenols of beverages has been done with tea and wine, and for that reason I try to keep up with what's going on in the tea field as well. The tea shoot, you know, the picked tea by the young bud and the two top leaves or whatever, at least the young flesh growth of the tea is picked to be the tea leaf that's used for the beverage. And this tissue is very high in phenolic content. On a dry weight basis, it may be more than 30% total phenolic materials. And as you might expect, the metabolism is very rapid in this tissue, and if it were only one way, only phenols being formed, then how do you account for the fact that there's less phenol in the older leaf? It has to come out has to be converted to other products in some degree. And when radioactive catechin was administered to tea plant shoots, up to 82% came out as carbon-14 CO2. So it was broken down, clear to carbon dioxide in a matter of 40 hours. And that's more and more impressive if you think that obviously, if our theories are meaningful, the radioactive material was metabolized the same way as the residual natural material, and considering that there may have been 30% of the dry weight, certainly in that general range, 10 to 30%, then this is a tremendous amount of metabolism. And not only that, but the tissue is normally accumulating phenols, so that if 80% is showing up as CO2 and yet it's still accumulating phenol, then there must be a tremendous turnover of these compounds, not only in rate but in amount. This isn't always so. Some also fairly recent work has shown that the turnover rate of a flavone in a certain flower, and again I've forgotten the reference and the particular flower. I tried to look it up this morning, but I wasn't lucky enough to find it quickly enough to get to the class. In any case, in this case, the half-life of Flavone turnover, marked Flavone administered, was of the order of 400 hours, which is not very rapid. So I don't mean to imply that the turnover rate of all phenols in all tissues is very rapid. Certainly lignins and tannins as a rule are fairly inert, but the point is they can be metabolized, and then some of the more rapidly metabolically changing compounds are very rapidly turned over. Does anyone want to raise any question or comment about the biosynthesis then before we go ahead and talk about the kinds of phenols in nature? Any comments or questions you'd like?
In this particular case with tea, if it's 30% of the weight, what is the function of these?
Well, to some degree we wish we knew. We'll be coming to some of the things we think are functions, but and we know of no reason for that large amount. It's idiosyncrasy of that particular plant as far as we know. Certainly they can have, just to give you an example, this high level has a lot to do with what eats the plant. For instance, it's been shown with deer browses that deer are very selective in what they eat, and they eat usually one or two kinds of plants at a certain time as far as the season of the year goes. And they select these largely on the basis of, or at least one method is flavor, and that has a lot to do with bitterness and tannin. So it may have been at one time a defense mechanism to keep bugs or creatures from eating them. Of course, now we've subverted that, and it's the very reason we do eat the tea or drink the tea from the tea plant. There are other reactions that we think are important. For instance, that high level of phenol would certainly give cold resistance compared to a similar plant without cold resistance, just from sheer physical water binding capacity. So that while the tea plant is not a plant that grows in areas where you have frost, on the other hand, it is among the plants that are very susceptible to chilling injuries, so that it may be that and tea is grown in rather, the better teas at least, in rather high altitudes, even though in the tropics, so that it may be related to cold response. And we'll be talking more about ideas in this regard, but when it comes down to a factual case, why is tea higher than the normal camellia? I suppose it is, I'm not sure, I haven't chewed many camellia leaves, but why it's so high, we don't really know. Any other comments or questions? Well, in discussing what phenols occur in nature, these notes that you have been used for some time, and for that reason they are somewhat out of date, but for two reasons I've hesitated to add to them. They already seem pretty formidable, and while there have been phenols discovered since these notes were made, they were pretty complete as of about 1968 or so. And at that stage, most of the type compounds had been found. The ones that have been found since then have mostly been rather exotic and unusual things, and so essentially what is being said is not really changed. Another reason is that the students tend to be frightened anyway, so I didn't want to add to the list very much or make it any more complicated. On your first page of this set of notes, there are 14 references listed, and the purpose of the references would be to in case you wanted to look up this sort of thing yourself and know where I got the data and maybe some further descriptions of any one kind of compound, these would be typical places to find it. The references 7, 8, and 9 have an asterisk, and probably 10 and 12 should have also. These were the tabulations from which I got most of my information. The most complete is Carr's of 1958, where he attempted to list all the naturally known plant substances in a typical German type of very detailed and conscientious tabulations that the rest of us have never learned to do nearly as well as the Germans have. And it's excellent as far as it goes. Of course, it's a 58 publication. And then Heilbronn's dictionary you might want to know about. It's one of the best places to find a compound. If you want to know what some named compound is that you never heard of before, chances are if you look it up in Heilbronn's dictionary, it will be there, and it will give you several references as to its properties and how its structure was shown and so on. The Merck Index, reference 10, was not starred because a new edition had come out since I made up the list, but what I did was, there's no tabulation in there, and what you have to do is just go through it. So I leafed through the whole blame thing and marked all the phenols, and then in the previous edition, and then added them to my list. I haven't had the courage to do that again for the new edition, but everything new that I've run across I've added to the list. And Robinson, Trevor Robinson's book, reference 12, is a fairly brief listing and structure discussion and importance discussion of classes of organic compounds in higher plants. Brief though it is, it's one of the best places to start because it not only talks about phenols, but alkaloids and fats and carbohydrates and what have you in a fairly succinct and yet pretty well done and as far as it was able to go, thorough fashion. We are trying to ignore as much as we can the nitrogenous, the alkaloidal type phenols, because mostly they've been studied as alkaloids. Nevertheless, many alkaloids have, in addition to a nitrogen group, which makes them basic and is therefore has been used chemically to isolate them and so forth. Many of them are phenolic. Nevertheless, most of what we're having to say about phenols could be applied to them without modification except for accounting for the presence of the nitrogen, and for that reason we have not included them on these listings, nor do we make any great effort to compare these compounds. By the same token, since steroids and certain other compounds that are more or less incidentally phenolic, you know, tremendous big structure and a little phenolic group on the end, we have tended to ignore those. So we're trying to talk about the about over 1,000 compounds that occur in nature as phenolics as their primary functional arrangement, phenolics or quinones. The 1,000 figure was based again on these earlier tabulations, and it might well be 1,500 by now, I'm not quite sure. But in that 1,000 we're not including any glycosidic derivatives of a readily hydrolyzable nature, or by and large we're not. Particularly glycosides, so that if you took a given anthocyanin, say malvidin, if you add in all the different named pigments that come from malvidin and are given different names because they were isolated from a certain flower or a different technique, then this would multiply that sort of total considerably, because many of the phenols are known as 3 or more glycosides with either a different sugar or a different position with the sugar attached. And of course these are chemically different compounds, so that then we get into really high numbers, say 5,000 or more of phenolic substances. But again, we intend to talk about the aglycones and not the phenolic function and not the sugar changes. Now, in trying to organize this material, it seems, once organized, it seems to me rather straightforward and simple, but perhaps we ought to spend a little time on page 2 so you'll see how we got this organized. We're studying, we've listed these by subgroups, and these may be to a degree artificial subgroups, but for instance, we start with the simple C6 phenols and quinones and then go to those with 1 carbon, 2 carbon, 3 carbons, and more than 3 carbons added to that phenolic ring, then come back to polycyclic phenols and so forth. And the classes that we will discuss are listed with Roman numerals near the bottom of page 2. Obviously we'll spend a good deal more time on certain classes, particularly flavonoids, and maybe a little on tannins and lignans compared to some of the others. Before we do that, I think we should point out that these now are what might again be called the organic chemist type compounds, in that they're isolated compounds, and their importance is not necessarily indicated. That is, importance in whether they have any commercial utility or whether they were just curiosities that were discovered by some chemist. So that some of them are not very important. They occur in very few plants, and unless you wanted to use them as a botanical marker for those few plants, then they wouldn't be even worth considering, perhaps, as for any purpose. Since the organic chemist tends to look for the unusual compound, and if we're just going to tabulate them, then we're giving equal weight to an important compound versus some that may be very rare. As a consequence, in the listings, you'll tend to have the compounds that I think are particularly worthy of note for some reason with an asterisk. And sometimes a question mark suggests that it might be worthy of consideration, or it might also indicate that the structure is questionable, but it usually means that it's worthy of consideration, but its natural role is a little less certain. There is a danger that in seeking the rarest compounds, you will find things that you're after. In other words, if you set out, say you want to publish a paper, say you're about to be offered a promotion as a professor, and if you don't have 3 publications, you're not going to get it, so you're desperately casting about for something you can publish. So you grab some plant and start isolating things from it. Naturally, you can't publish it if you find chlorogenic acid in it, because you'd expect to find that in everything. So you've got to find something exotic. And there are some very horrendous examples of this in the literature. I won't give enough details that you know, but there was, for instance, a student that was in this class a few years ago who had done his thesis research in another country, and one of the things that had been found was catechin glycoside. Well, catechins don't ordinarily occur as glycosides, and I had occasion to review this student's thesis, and I was quite convinced he didn't prove that it was a catechin glycoside either. And in fact, he wasn't even aware that they were rare in nature, and I think he was misled by his professor, probably for the kind of reason I mentioned. So even so, you'll find this published in the literature as I think the only instance of a catechin glycoside found in nature. And unless you went back to the thesis and talked to the people, you wouldn't know that it probably was not proven that that's what had occurred. Vice versa, the chemist tends to find the unusual compound. The botanist tends to find only the expected compound, because the botanist is usually working with paper chromatography or some similar technique, and he has a pattern of spots, and he says, oh yes, that must be catechin, and this must be chlorogenic acid, and so on, because the RFs are in the right position, and so he publishes the paper and says, this is what they are. Well, he's just as apt to be wrong. If it's a similar but different compound, he'd never know the difference, as long as the RF is about the same, the color reactions are about the same, as they usually or frequently can be. So that you have both errors, finding only the common ones when something else is there, and this 2-hydroxyflavanone that the long sheet that we were passing out, they're not 2-hydroxy, but alpha-hydroxychalcone type derivative that we were saying was only recently being proposed as a biochemical explanation of how the hydroxylation in the heterocyclic ring may go. These compounds have only been found in about the last 2 years. And yet, now that they've been found, everybody's finding them in lots of things. So it's another example of where they were obviously detected before, but were assumed to be something that they already knew and nobody investigated. So both of these do occur, and you must keep in mind that that is a problem, particularly in your own research if you're doing this sort of thing. Another point is that rarity is a function of amount as well as presence, because a compound that may be present in almost everything, if it's present in such a tiny amount that it's difficult to study and other similar compounds overshadow it, then you may miss learning about it or detecting it. And for instance, pyruvic acid again is a pretty good example. It's no doubt it must be universally present, knowing what we do about its biochemical importance. Yet if you try to find pyruvic acid in most tissues, you're going to have a whale of a time getting enough out to be sure it's there. In some instances, some certain plant or some certain tissue, some certain time of the year, a rather rare phenol may make up a tremendous percentage of the dry weight or carbon content of a certain plant and a certain tissue. So even though it's ordinarily unimportant, it may be massively important in that particular case, and the tea that we just discussed might be such an example. Now, we can divide phenols roughly into two groups. I mean, there are many ways you can divide them, but if we divide phenols into two groups in plants, this is quite useful in the sense that there's one group that's almost universally present in higher plants, and there's another group then that is more rare and exotic, and only certain plants have certain members of that group. So that the anthocyanins of the common type, the catechin, epicatechin, and close relatives, the flavones of certain kinds, the particularly flavonols of certain types, quercetin, myricetin, apigenin, these would be commonly found in almost all plants. Yeah, thanks. Would you try to close him off if you can? On the other hand, anthraquinone or some derivatives other than the cinnamates and flavonoids of common types are not very widely found, and some of them are unique to a certain plant. So the gossypol is a toxin found in cottonseed and only in certain species of cotton, much less cotton is in the hibiscus family, and the cotton that we get cotton from is one of the few plants of the close relatives that has gossypol in it. And just why that is, we can speculate on later on. But this sort of thing governs what is found, and even though something is rare, it may be critical in certain instances. Of course, an extremely rare phenol that only occurs in one plant can have no chemotaxonomic importance. In other words, if it's only in that plant, well, then if you see that plant, you know it's there, and so the identification of that phenol wouldn't help you find that plant because it's only one. But in other cases, you'll find that the distribution is quite useful, and phenolic distribution is one of the best chemotaxonomic clues as to how plants ought to be classified. And this is becoming more and more used in that we've pretty much exhausted the comparison of the physical features of the plant and so on. And if you run into any anomalies that you'd like to clear up, then the chemical constituents is the next area for examination to see how these plants ought to be included or separated botanically. Another point is that when one sets about identifying these compounds in nature, you use fairly severe techniques, and the isolation may be reasonably delicate, but not always. It depends on the technique used. So you may produce artifacts. So you may find a phenol that you think is unusual, but in study it may turn out that it was made from a more usual phenol by the particular isolation technique used. And this is even more likely if you have to degrade the compound. In other words, if it's a glycoside or some larger unit compound, a polymer or something of that sort, then if you're breaking it down to subunits, and some of these subunits seem unusual, it may be because you've made them so rather than that they occur in nature in that condition. So you should be aware of artifacts. I know of one good example that I don't remember the complete details on, but there was a case where in the poplar tree system, there was one poplar tree, I think a European poplar as compared to an American poplar, where a certain phenolic substance was supposed to be in one and a related but quite different phenol in the other. And it was then later found that the only difference between the two was that they'd use different isolation techniques. They'd use a lead acetate precipitation in the European studies, and that had caused an isomerization of a double bond, and in fact the same compound occurred in both. So that this is one example of how this can mislead people. And a lot of this sort of thing appears in the older literature where it's a one-paper deal. In other words, a certain rather unusual plant is studied for the kinds of reason already mentioned. Somebody wants to do something he can publish. He picks out the most esoteric thing he can find that nobody else has worked on, and nobody else wants to work on it again, so that it gets published, it gets in the literature, and then it's assumed to be right. Well, it isn't always, although usually it is. I'm not impugning morals and ability of all scientists. On the other hand, they're just as human as anybody else, and you can find this sort of thing fairly frequently, and you need to be aware of the possibility. Another problem is that if we consider the published structures on these compounds, we of course will get some clues as to biosynthesis patterns, and this will give us some clues as to how these compounds were produced. Not only which shikimic or acetate or whatever pathway, but also maybe which monomeric phenols were used to make what larger phenols, so that we could see within lignin structures some guaiacol units which suggest excuse me, suggest ferulic acid connection, and in fact it can be shown that ferulic acid can be rather rapidly and without rearrangement of carbons incorporated into lignin. So just by looking at some of the units from the compounds can give you a good deal of information sometimes, and that's one reason we discussed, however briefly, the biosynthetic relationships before coming to the nature of the compounds. Well, that's a lot of introduction. Let's take a look very quickly at some of the classes, and we will make it quickly. What I would like for you to keep in mind as we go through, as I already suggested, you should leaf through, look at the classes, read the observations, maybe look at some of the structures. Now, the structures are not generally drawn out for you. If they're complicated, they are, but they're all given on the basis of a phenol, at least in the earlier cases. So we're talking about this compound, and then this being considered the one position. If it says 2-methoxy, that means that we're talking about the methoxy in this place. If it says 4-methyl, we're talking about the methyl in this position, so that even though the structure isn't drawn for you, if you think of this as the one carbon, then you'll be able to see the structures we're talking about. I suspect most of you aren't terribly great botanists. I certainly am not. But also I'd like for you to sort of glance on the right-hand side of each page where it says the plants that these compounds are found in. And the number in parentheses is the number of genera reported to contain that compound, so that we're trying to give you some idea of how frequent or how rare the compound is. So that, for instance, apionol does not occur in nature as far as we know, but it's been found as a degradative fragment of apiole from parsley, so that it's a natural compound as a unit within another unit. Well, these are the sorts of things we'd like for you to look for, and as I say, we'll skim through at least most of these very quickly. For instance, in this plain C6 phenols and quinones, all possible isomers of the 1 through 5 hydroxybenzenes occur in plants. The 6-hydroxybenzene has not yet been found in nature. They are not, however, usually present as simple free compounds. If they occur as the simple compound itself, they're usually in a glycosidic or ether or both form, sugar attached or partially methylated. Methyl ethers are common in this group. Methylene ethers are fairly common, and other kinds of ethers are rare. As a rare example, the ethoxy ether of hydroquinone is found in Elysium, and I've forgotten what Elysium is. I think it's a lily. At least it's in the lily family, but I'm not sure which one. Some of these generic names may not give you too many clues, but Pinus is, of course, pine. So Pinus Sylvestris is a pine species. Ephedra gives us ephedrine, and you know of the drug at least. And guaiacum is a plant which gives us the name for guaiacol, and so on. Some of these I'll mention, some of them I think you'll recognize, like sequoia, and allium is onion, or the onion family, onion genus, so that some of these you'll recognize, and quite a few we've given the common names to as well. You might wonder what quino is, or kino as you prefer. It's pronounced both ways. This is from eucalyptus trees, and it's apparently the result of a kind of injury, although the injury evidently occurs naturally, so that you can't always define what the injury was. And I haven't actually seen this, but in, say, the trunk of a eucalyptus tree connected with some sort of injury or natural splitting site, there are pockets, and some of these pockets will contain up to several gallons. I've heard of these as much as 30 gallons of a very tannin-rich aqueous solution, and this tannin-rich aqueous solution is called quino or kino, and has been used to tan leather and so on. So that's what that material is, and it's in the eucalyptus. Well, the observations in connection with the simple phenols, besides the fact that all of the 1,5 OH isomers are present, is that the only glycosides that have been found in this case are glucosides. Now, I'm not certain that's still true, but it's certainly still largely true. And it's interesting that glucose is, by all odds, the most common, but not the exclusive kind of sugar that's in all phenolic glycosides. And in fact, there are sugars that are found in glycoside form that are not found any other way either, for that matter. So that rutin, for example, is a flavonoid, a quercetin derivative, which has rutinose, which is a sugar composed of glucose and rhamnose. The rhamnose is not unknown otherwise, but it's most commonly found in that particular compound and close relatives. So there are sugars that are unusual, but glucose is by far the most common and is exclusive in this particular category, apparently. The hydroquinone distribution in pear and its relatives has some taxonomic significance. If you realize that we're in the wing of the building devoted to pomology, so it's appropriate to talk about pome fruits, and apples and pears are pome fruits, and yet hydroquinone derivatives are found in pears and different compounds in apples. And there are genera within the types and species within the types that do differ in ratios and so forth. The quinones simple quinones tend to be in fungi, and the phenolics tend to be in higher plants. We could make one side excursion in this particular group. Sometimes, in obscure cases, there's important relation to these compounds. For example, in the cocklebur, which you may know, this plant in its seedling stage, it's a dicotyledon, in the cotyledonous stage, which looks about like so, and it comes up very early in the spring when very little else green is present in the pastures in a cold climate. And if animals eat this, then at this stage, they get a tremendous dose of hydroquinone. And the hydroquinone is sufficient to kill the animal, so that loss of pigs and sometimes ruminants from this kind of cocklebur sprout is a fairly widespread agricultural problem, at least for those subsistence animals that have to graze to live and have to eat something in the spring when there isn't much yet growing. It's less common today than it used to be, but it's still a problem, and it's less common largely because we pen up our animals more and bring the feed to them instead of letting them go get it. I think we're going to have to reverse this tendency one of these days when energy and so on gets sufficiently short, so it'll probably come back to haunt us even more. Well, that's a sidelight. That covers what I want to say about these compounds. You might want to remember, if you don't already, the asterisk structures, that is phenol, catechol, hydroquinone, resorcinol, pyrogallol, and chloroglucinol in that group. The C1 substituted phenols include paracresol, and it occurs in ylang oil. It's an exotic perfume oil, and a few other places. Orsanol occurs in certain materials. It's got sort of a sweet but unpleasant taste, phenolic medicinal type on top of sweet. These occur, and although they're asterisked, they are the most interesting and most informative in that group. They're not all that important in general, and not compared to the simple ring compounds. There is one point I might make here. Orsanol can occur in 3 isomers. And in the third position. So you can see that here this position would be equivalent to substitution in this position, so there's really only one compound. This one is called the gamma form, and this one is called the alpha form, and this one is called the beta form. And that type of nomenclature is carried on in certain other compounds with this same kind of isomerization possible. So if you run across these, I thought you ought at least have heard what it was. Now carrying on from the C1 group, which is relatively limited and generally only found as a glycoside, only one known, and that was an orsanol beta D-glucoside. I've forgotten which orsanol. I think it's the alpha form that occurs most commonly in nature. These appear to be generally shikimic derived. If you notice the phenolic, wait a minute. Yes, these appear to be generally shikimic derived, except that you would expect this to be acetate derived with the meta arrangement of the OH. In other words, we would generally find both possibilities, and in the higher plants the substitution tends to emphasize the shikimate derivatives. In lower plants and fungi, lichens, tends to emphasize the acetate type structure. The C2 group is less common, and no glycosides, very limited, mostly appear to be shikimate derived. The allyl group with the propyl side chain, but with the double bond in the terminal position, is a fairly widespread group and includes eugenol, which is the clove-related material, and also found in pimento and bay oils. These compounds are quite important in flavor and essential oil characteristics, whereas the propenyl derivative with the double bond between the 2 carbons nearest the ring, these compounds are not nearly as frequently found, although they are known and do occur in nature. And isoeugenol is the difference between the eugenol and isoeugenol is the movement of that double bond. Birch has proposed a mechanism which is pretty much paper chemistry at this stage, but he's proposed a reasonable mechanism as to why this structure is more prevalence, and it illustrates a couple of interesting points. He says that this structure is made by the linking of, well, first it would be a synamic derivative, as you would expect. This chalk is getting imaginary. And the synamic derivative is reduced to the alcohol group and then phosphorylated. But the double bond would still be in the position that one would expect for the synamic derivative, and we know that occurs because we have alcohols of this series. And then apparently we have an equivalent of a negative unit electron attack, which would amount to reduction, and the reduction would be via an NADH type reduction, and that causes the electron insertion at this point, which would cause the electron migration in this direction, and you would end up with the phosphate, to balance it, we'd go that way, end up with eliminating the phosphate from this end and moving the double bond to this position. So the net result in terms of side chain would be this, where the ring is then attached here. So that this is, according to him, and it seems the most reasonable, the reason that the allyl side chain seems to be most common. And also would help to explain how one eliminates, this started out as a carboxyl. Well, you can reduce it to the alcohol, but how do you eliminate the oxygen? Well, apparently you do by carrying it away as a phosphate unit. This, I say, is somewhat speculative, but it seems to be a reasonable explanation. On the other hand, then, one having the allyl function can isomerize back to the propanol derivative, which has the double bond in the other position, by treatment with alkali in the test tube, and apparently this sort of isomerization can occur in nature by less drastic means. So it would appear that by and large the allele is what is produced, it's most commonly found, and then by isomerization it can convert back to the propanol. Well, obviously, I'm a minute over. I hadn't been watching too closely. We'll have to quit at this point. We'll skate through the rest of these quite rapidly in a similar fashion, and see you then